Hello everybody, this is Matthew Becar and I will be presenting on the evolution of antibiotic resistance in bacteria. What are antibiotics? Antibiotics are a class of molecules which kill or inhibit microbial organisms with disregard to the source of the target. In short, they are used to treat bacterial infections. The use of antibiotics in a variety of settings has led to antibiotic resistance. Penicillin, for example, was discovered in 1928, and in 1940, before it had even been used as a drug, researchers discovered a bacterial penicillinase causing antibiotic resistance. We could see the reemergence of diseases that have long been treatable with increased bacterial antibiotic resistance. We will search to understand how bacteria evolve in order to become resistant, the mechanisms of gene amplification, hypermutable bacteria, the SOS pathway, horizontal gene transfer, and superbugs will be explored, and their impact on the evolution of antibiotic resistance in bacteria investigated. The general mechanism of bacterial evolution is quite simple. Antibiotics provide a selective pressure on bacteria. The most resistant of these bacteria will survive and go on to produce offspring that are more resistant than the previous generation. If you remember, evolution is a change in allele frequency over time, with each successive population becoming more resistant. The first mechanism of evolution we will be discussing is gene amplification. Gene amplification is the duplication of one or more genes on the chromosome of a bacterium. Duplication of a gene can cause overexpression of the protein which that gene encodes. Streptococcus agalactiae is normally susceptible to antibiotics, such as sulfonamide and trimethoprim. However, there is a strain called CCH290 that is resistant. In this bacteria, this resistance is caused by 13 gene amplifications. Amplifications of the full genes, such as full P, are especially important in the resistance of this bacteria. Full P encodes a protein called dihydropteroate synthase. This is the target of the antibiotic sulfonamide. In this graph, in the highlighted area, you can see the strain CCH290 and its expression of full E, full P, and full K, as well as in the column under ZD, or zone of inhibition, the amount of inhibition in this bacteria. There is a much smaller zone of inhibition in CCH290 compared to other strains of this bacteria, suggesting that it has become extremely resistant compared to the other strains. This is correlated with an increased expression of the full genes. The next mechanism of evolution is hypermutable bacteria. Selective pressure of antibiotics may select for bacteria called hypermutators. Hypermutators are a class of bacteria which have an increased frequency of mutation. This selection occurs because bacteria that rapidly mutate are more likely to develop resistant strains through mutating that will be selected for and increase in frequency over generations. This is known as genetic hitchhiking. An experiment done by Antonio Oliver shows that populations of bacteria which are exposed to a continuously changing environment often generate mutators. A highly variable environment is the perfect condition to stimulate hypermutators, as those bacteria which are able to adapt and evolve most frequently and best will be the most fit. In this bacteria, Pseudomonas aeruginosa in cystic fibrosis patients was studied. In cystic fibrosis, the lung mucus layers become more viscous and have increased osmolarity, creating an ideal environment for bacterial growth. The disease causes the lungs to lose some of their ability to fight against infection and provide an ideal site for this bacteria to grow. You can see the mutation frequency in CF patients versus non-CF patients, CF representing cystic fibrosis. CF patients had a highly increased mutation frequency. Hypermutators not only become resistant to current methods of treatment, but are also more likely to become resistant to future treatment with novel antibiotics. Horizontal gene transfer is a potent mechanism of evolution within bacteria. This refers to the transfer of genetic material from one organism to another. Sometimes the transfer of genetic material will contain a resistant gene. This can lead to antibiotic resistance. Horizontal gene transfer occurs very frequently in nature and is most likely a large contributor to the evolution of many bacteria. Treatment with antibiotics increases horizontal gene transfer in some bacteria, leading to increased levels of resistance, especially when non-lethal doses are used. For example, Vibrio cholerae increases the transfer of a gene encoding resistance to a few antibiotics when in the presence of ciprofloxacin, an antibiotic. The SOS pathway is a response initiated when bacteria have DNA damage. This happens in all bacteria and is universal. Some antibiotics induce the SOS response in certain bacteria, which increases the rate of mutagenesis. Increased mutagenesis increases the likelihood of resistance evolving via mutations. Induced SOS response showed a drastic increase in the levels of horizontal gene transfer as well. Those antibiotics that target DNA replication and repair have been particularly potent in inducing the SOS response and have greatly increased horizontal gene transfer rates. Superbugs are bacteria that aren't treatable with antibiotics. They are resistant. Superbug antibiotic resistance can arise through various methods, such as the ones we have discussed before, and more. One cause of increasing resistance is that around 50% of all the antibiotics prescribed for the treatment of infection are not properly prescribed, meaning they have incorrect dosage or length of treatment. In the presence of antibiotics, the most mutated and resistant bacteria survive and reproduce, passing on their mutated genes vertically and increasing the resistance of the bacteria. When an incorrect dose is prescribed and the antibiotics select for the mutated bacteria, but do not eliminate the infection, the bacteria become more resistant and harder to treat. Horizontal gene transfer, or of resistant genes from organism to organism, can increase the level of antibiotic resistance within bacteria. The use of antibiotics to treat bacterial infection itself has increased bacterial resistance by pushing the bacteria to suitable strains which can better adapt and cope with the pressures of antibiotics. Gene amplification can lead to increased resistance due to increased protein expression. The SOS pathway can be activated by many antibiotics, and the increased mutagenesis and horizontal gene transfer levels can cause the evolution of antibiotic resistance. Superbugs are created by the previous mechanisms as well as many others, and strict regulation of the prescription and dosage of antibiotics should be taken to prevent the spread of superbugs. As you can see, there are diverse methods and mechanisms of evolution that cause bacterial antibiotic resistance. We must be careful in our prescription of antibiotics. More research is needed to find different mechanisms of antibiotic resistance. As we continue to research and find more mechanisms by which bacteria are able to mutate and evolve into resistant organisms, we will better be able to understand how we can stop this and how we can increase our treatment of these bacteria. More research could be done into ways of how to possibly find a way to change resistant bacteria back into non-resistant bacteria. We're excited.